Om Swastiastu. We are from SGD B01 PSSKPD 2020 first semester. In this occasion, we're going to present our student project entitled Central Dogma in Molecular Biology. Here's the members of our group. My name is Winnie Chandra, and I'm going to explain the background. The central dogma of molecular biology was formulated by Francis Crick in 1957, and it is called the central dogma because it started with the sequences of amino acids that create the proteins and are encoded in DNA. It is divided into three phases, which are DNA replication, DNA transcription to RNA, and RNA translation to proteins. Next is problem formulations. The first one is how is the structure and characteristics of DNA. The second one is how is the structure and characteristics of the four types of RNA. The third one is what is the definition and process of central dogma. And the fourth one is what are mutations. Next is the purpose. The purpose includes to describe the structure and characteristics of DNA, to describe the structure and characteristics of the four types of RNA, to describe the definition and the process of central dogma, and to describe what mutations are. Next, the benefits are give general and specific knowledge about central dogma, disclose the specific structures and characteristics of DNA and RNA, and fully perceive about the mutations. Next.
My name is Putu Dewi, and I would like to explain about DNA. A DNA molecule occurs of two long polynucleotide chains structured of four varieties of nucleotide subunits. Each of these chains is called a DNA chain or a DNA strand. Each of these nucleotides consists of one 5-carbon sugar, which attached to one or more phosphate groups and a nitrogen embodied base. The sugar in the nucleotides is a deoxyribose attached to one phosphate group, and the base are adenine, cytosine, guanine, and thymine. The structure of DNA is three-dimensional, which is called double helix, which is the result of chemical and structural features of its two polynucleotide chains. These two chains are held together by hydrogen bonds, forming a base pair, where guanine forms a base pair with cytosine while adenine with thymine, which run anti-parallel within the DNA molecule. Next, DNA carried genetic information in the linear sequence of nucleotides. Those genetic information stored in DNA are called its genome, and that carries all the proteins needed to synthesize. Next.
My name is Williem Alexander, and I'm going to explain about the structure and characteristic of RNA. RNA is a ribonucleotide purine and pyrimidine polymer that is connected each other by 3,5-phosphodiester, which have three parts such as 5-carbon sugar, and phosphate group. RNA has ribose as sugar and 4 common nitrogen bases such as adenine, guanine, cytosine, and uracil. Pyrimidine uracil substitute thymine of DNA. Guanine pairs with cytosine and adenine pairs with uracil. RNA has hydroxyl groups at the second positions of the ribose sugar, so helix can adopt A-form geometry rather than B-form geometry and makes RNA more flexible if we compare to DNA. Messenger RNA or template RNA, transfer RNA, and ribosomal RNA are among the three major classifications of RNA.
Hello, my name is Dewi, and I will explain about the central dogma and its processes in general. The central dogma of molecular biology is a statement that explains how the information from the nucleic acid, DNA and RNA, flows and translated into protein. In 1958, Francis Crick was the first man that stated this statement. Nowadays, the central dogma process has been explained as a progression that starts by the transcription of DNA or deoxyribonucleic acid to ribonucleic acid or RNA, and continued by the translation of RNA to protein. Next, this genetic flow has formed many biological processes such as cell division, signal transduction, and also the homeostasis of organisms. Next.
My name is Karina Nityani Kumar, and I will be explaining about DNA replication. DNA replication happens in all creatures and take into account hereditary legacy. Without replication, when the cell isolates, the data will be part and just halfway passed on. The cycle of replication permits cells to create new hereditary materials, or DNA, utilizing unique DNA as a layout. Next, the cell cycle comprises of 4 stages, G1, S, G2, and M. During the G1 stage, cells develop and produce material like nucleotides, forerunners as groundwork for DNA replications in the S stage. Replication happens in the S stage cell, and new hereditary material is incorporated as a groundwork for the cell division. Next, replications in prokaryotic and eukaryotic cells is exceptionally similar. The prokaryotic DNA is round and in this way has just one purpose of inception, where replication starts and moves in bidirectional way, while eukaryotic DNA replication happens in the core of a cell, where a new DNA is made utilizing the first DNA as a format. That is all from me. Next.
My name is Nindya, and I will explain about transcription. Transcription is a process of gene expression that uses DNA as a template to synthesize RNA. It occurs in the nucleus of the cell. Not all of the part of DNA will be transcribed, but only certain parts called transcription unit. It consists of promoter, RNA coding region, and terminator. The transcription process is catalyzed by an enzyme called RNA polymerase. In eukaryotic organisms, there are three different types of RNA polymerase to catalyze the synthesis of different types of RNA. Next. Transcription consists of three stages. They are initiation, elongation, and termination. Initiation is the stage when the RNA polymerase recognizes the promoter region of DNA. Elongation is the stage when RNA polymerase travels along the DNA, adding ribonucleotides that pair with the DNA template. And termination is the stage when the terminator region is transcribed. That's all for me. Next.
My name is Mira, Wrycza, and I'm going to explain about translation. Translation is the process where the cell converts information in an mRNA molecule into a protein molecule. There are three stages. First is initiation, where the first tRNA is attached to the start codon. Second is elongation, where the amino acids are combined into a polypeptide chain. And termination is when the finished polypeptide chain is released. Next. During initiation, the tRNA that carries the amino acid methionine binds to the start codon of the mRNA sequence. The large subunit of the ribosome binds to create the P and the A site, and the first tRNA occupies the P site. The second tRNA enters the A site. The methionine is transferred to the A site, and the first tRNA exits, and the ribosome moves along the mRNA. Then the next tRNA enters. Each mRNA is read one codon at a time. Termination happens when a stop codon enters the ribosome. That's all from me. Thank you.
My name is Dewa, Fony, Prema, Shanti, and I would like to explain reverse transcription. Reverse transcription is a change from RNA to DNA with the aim of producing complementary DNA. Reverse transcriptase uses an RNA template and a short primer complementary to the 3' end of the RNA to direct the synthesis of the first strand cDNA, which can be used directly as a template for the polymerase chain reaction. Reverse transcriptase, also called RNA-directed DNA polymerase, is an enzyme encoded from the genetic material of retrovirus that catalyzes the transcription of retrovirus RNA into DNA. As for the benefit obtained from reverse transcription, mechanisms are described. The transcription process, the testing of genetic tumors becomes easier and more. That's all from me. Thank you.
Hello, my name is Darren, and I will explain about mutations and mutagens. Next, mutations are caused by many factors, such as physical or chemical agents called mutagens. These agents could cause mutational effects on genes. One example of physical mutagens is X-ray radiation. In the 1920s, Hermann Müller conducted a research using X-ray radiation and discovered that it induced genetic changes. Through this research, it is implicated that high-energy radiations are dangerous to living organisms. Another example of mutagenic radiation is UV light, which could disrupt thymine dimers in DNA. Aside from physical mutagens, there are also chemical mutagens. There are several categories of chemical mutagens. The different categories depend on how the mutagen works. Some chemical mutagens disrupt DNA replication, while some others insert themselves into a DNA strand and alter the structure. Researchers determine the mutagenic nature of chemical mutagens by testing their ability to cause cancer, since most carcinogens are mutagenic. That is all for my explanation. Next.
Hello, my name is Sendi Aprilio and will explain about types of mutation. Based on a scale, mutations divided into large mutations that occur in the chromosomes and small mutations that occur in the DNA or RNA. Next. Small mutations happen more common. This mutation causes changes in nucleotides so the codon change and affects the protein. This mutation consists of substitutions and insertion or deletion. Substitutions is a mutation that replaces one pair of nucleotides with another one. For example, DNA template CCG becomes CCA because guanine is replaced by adenine. So the codon that's supposed to be GGC changed into GGU, but the amino acid still glycine. This mutation called silence mutations because the changes do not affect the amino acid. But there is also mutation that affect the amino acid called missense mutations. There is also nonsense mutations that change codon into a stop codon, so the protein formation ending prematurely. Meanwhile, insertion or deletion is mutations that adding or subtract one or more pairs of nucleotides. This mutation having severe effects because a shifted framework will affect the reading process and amino acid that form will be completely different. For example, in insertion, the codon change because guanine is added, so the protein become leucine, leucine, cysteine, and threonine. Same effect also happen in deletion. The codon change because guanine is subtract, so the protein become phenylalanine, tyrosine, and histidine. That's all from me. Thank you. Next.
Hello, my name is Gede Sutha Samudra, and I will explain you about the example of mutations. Most dangerous mutations are mutations that cause large changes in protein. For example, changes in single amino acid that crucial in the beta globin subunit in hemoglobin that can cause sickle cell anemia. Next, sickle cell anemia is a genetic disease where red blood cells can take the shape of a crescent or sickle, and that change allows them to more easily be destroyed, causing anemia among other things. Sickle cell mutation is a non-conservative missense mutation that results in the 6th amino acid of beta globin being a valine instead of glutamic acid. A non-conservative substitution means that the new amino acid, valine, which is hydrophobic, has different properties than the one it replaced, glutamic acid, which is hydrophilic. Replace cycling of blood cells damages the cell membranes and promotes premature destruction. Since this happens within the vasculature, it is called intravascular hemolysis. This destruction of red blood cells not only leads to anemia, which is a deficiency in red blood cells or loss of the normal levels of the hemoglobin, but also means a lot of hemoglobin spilling out. That's all from me. Thank you. Next.
Hello, my name is Bawa. I would like to conclude our presentation. DNA is made up of two long polynucleotide chains that are structured from 4 types of nucleotide subunits, each of which is called a DNA chains or DNA strand. RNA is a ribonucleotide purine and pyrimidine polymer that is connect each other by 3,5-phosphodiester, which have 3 parts such as 5-carbon sugar, and phosphate group. Next. The central dogma of molecular biology explains how the process of reading genetic material into proteins play a role at every metabolic stage in an organism. The central dogma of molecular biology consists of several processes, including DNA replication, transcription, translation, and reserve transcription. Next. Mutation is genetic information in organism can undergo change due to certain condition. Mutations are caused by many factors such as physical or chemical agents called mutagen. That's all from me. Next. As you can see, here are some of the references that we use. Next. This is the end of our presentation. Thank you for your time and attention. We apologize if we make mistake intentionally and intentionally. We will close this presentation with Paramo Shanti. Om Shanti, 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 Shanti Om. Thank you.